Okay, so to start off, I have this diagram here showing the visual sensory pathway. So here is sort of looking down on the subject, and here is sort of looking at a subject, looking at the side of it. So you can see superior and inferior. Just to go over it really quickly, your temporal visual fields, so what your eyes are seeing, cross to hit your inner nasal retinal fields, and then vice versa here, going out from nasal to temporal. So you're going to use the retinal fields when you're deciding what's going to cross going further back. So with that retinal field, you have your temporal outer retinal fields are going to stay on the outside when they go to the lateral geniculate nuclei. But then your medial retinal fields are going to be the ones that cross in the optic chiasm right here. And I've kind of drawn the lines far apart from each other to illustrate what's crossing and not crossing, but the reality is that these outer ones are going to follow pretty closely and just curve and not cross. And that's important because, as you know, in front of the optic chiasm, or before it crosses, these are going to be your optic nerves. And then after the optic chiasm, so here down and then about here down, is going to be your optic tracts. So just keep track of that. And then once you hit the lateral geniculate nucleus, you're going to have your upper visual fields or your lower retinal fields taking Meyer's loop, which is going to go into that temporal lobe and curve. And then the rest of your geniculocalcarine tract that's just not named is going to be your lower visual field / upper retinal field. Meyer's loop from the lower retinal is going to stay low and end up hitting the lingual gyrus underneath the calcarine fissure. And this is going to stay high and hit the cuneate gyrus. And so that's better illustrated with this one. So remember, your superior is going down, inferior is going up onto the retina, and that's what stays high. So when I say stay high, I'm not talking about your visual fields. I'm talking about your retinal fields. And it's just going to cross just like that. Now here is this deficit picture we've seen a million times, but just to hit the highlights, you cut an optic nerve. All the information that this nerve has so far is just from this eye, and it's got everything from this eye. So you cut it, you get monocular vision loss right here. If you cut the optic chiasm, what was crossing was our medial or our nasal retinal fields, which is going to end up being our temporal visual fields. So when we cut those off, it's like we get blinders put on. So that's bitemporal hemianopia. Then you go lower. If you cut the optic tracts, you're going to have the outer retinal field of the ipsilateral eye that hasn't crossed, and you're going to have the nasal retinal field of that contra eye that has crossed over to join. So you're going to lose from the optic tract your ipsilateral temporal retinal and your contralateral nasal retinal. And when we translate that to your visual fields, it's going to be the opposite. So basically, when you cut one optic tract, you're going to cut the opposite side's visual fields in both eyes. So that's why it's going to look like this. So you cut a tract, you're going to cut the other half of your vision out on each eye. And then coming down here, if you cut all of your geniculocalcarine tracts, which includes Meyer's loop, so if you cut everything, you're also going to get that same damage because it's just like you're cutting the tract. You're cutting everything just beyond it. However, if you just cut one or the other, you're going to only get a part of this contralateral visual field loss. So your Meyer's loop is carrying your upper visual fibers. So imagine just only losing that top part right here, sparing the bottom because you didn't cut this geniculocalcarine tract. So that's why you get Meyer's loop pie in the sky. But then if you did cut just this one and spared Meyer's loop, you'd have the exact opposite. So again, when you're cutting one side of the visual system, typically what you're losing is going to be the opposite-sided visual field. So just kind of keep that in mind. And it's not on this picture, but if you imagine that when these are going back to that occipital lobe, when you're going to your visual cortex, if you were to just take out the right-sided lingual gyrus, you have to think about what fibers were traveling there. So that was going to be Meyer's loop. So if you take out the lingual gyrus on the right side, you're going to get the same deficits as if you cut it right here. So you're going to get that pie in the sky. And so just keep that in mind when you think about injuring specific gyri. Okay, now to dive into the lab stuff specifically, we've got some pretty quick, easy ID to start. So as I was showing you in that diagram earlier, your optic nerve is going to be what's coming from your eyes. It's been cut here. So your optic nerves are before your optic chiasm, where it crosses right there, and then continuing where you can't quite see is going to be your optic tracts going towards your lateral geniculate nuclei. And then your infundibulum is your pituitary stalk, which is going to be right here, if you remember that. So this is sort of why when you get pituitary tumors, a lot of the time you can have vision loss because you're sitting right here next to all of your visual apparatus. Even that oculomotor nerve is sitting right there. Okay, now to dive into this cross section, first let's sort of analyze where we are. We see we are in the midbrain because we have our pes pedunculi / our crus cerebri right here, substantia nigra right behind it. What is that producing? Dopamine. And I feel like this is going to be a question, so heed my words. The pars compacta, which is the pigmented part of the substantia nigra, is dark because there is neuromelanin being produced. And so that's what's giving it the dark pigmentation. You can't see it on this sort of stain, but you know the pars compacta is pigmented. Neuromelanin is produced as a byproduct of the reaction of tyrosine being converted to dopamine via the enzyme tyrosine hydroxylase. So take it or leave it, but that's just a fun little fact there. Here you have your red nuclei, and up here we have some colliculi. Can you tell if it's superior or inferior? It's superior because it's written right here. But you can also tell just because the superior colliculi tend to actually sit closer together than the inferior do, and also because the red nucleus is so obvious here, you can tell that you're high up enough that it's superior. If it was inferior colliculi, your red nuclei wouldn't be quite as prominent and perfect with the edges here. So we have our median geniculate nuclei right here, which means that lateral to that is going to be our lateral geniculate nuclei, a lot bigger on this side, you can see. And then we just talked about the visual pathway, so the optic tracts are feeding into the lateral geniculate nuclei. So then it would make sense and follow that this pathway and tract coming right here to the lateral geniculate nucleus is therefore the optic tracts. So right here and right here. And then this is part of your thalamus, I'm pretty sure, but I don't think that's going to be super important for this exam. Okay, so now we have a mid-sagittal cut of the brain. So we've seen this a million times, but let's just add in a couple things we need to be paying close attention to. First off, our calcarine sulcus. We've talked about this in the visual pathway. It is what's separating right here your occipital lobe, separating your lingual gyrus from your cuneate gyrus. Your visual cortex, area 17, is within the depths and banks of this calcarine fissure. So just this visual cortex is right here in the occipital lobe. Yeah, short and sweet. Okay, now we're going to move on to some different types of cuts here to illustrate the optic radiations. So these radiations are what is leaving from your lateral geniculate nuclei to travel to your visual cortex. So that's your geniculocalcarine tract, which includes that subdivision called the Meyer's loop. So what you're going to see is here's your occipital lobe, and so your visual cortex is going to be around here. So your optic radiations are always the lighter colored bands traveling towards it right there. So this white strip is going to be traveling from your lateral geniculate nucleus back to that occipital lobe. You can see it here really well because of the striations. It helps you visualize that pathway a little bit better, just traveling from that general LGN area back. And here's one more good visual of it when we've got the myelin stain going, traveling just straight back and radiating from the LGN to the visual cortex. Okay, so now we are back to the bottom of the brain. We're going to just really quick again point out the optic nerves crossing in the optic chiasm. going and continuing as your optic tracts. So your optic tracts are feeding to your lateral geniculate nuclei, and you can actually see those on this gross specimen. So when you follow this tract, you end up seeing this bulbous structure. Oh my goodness. Well, that's your lateral geniculate nuclei right there. And then what is leaving the lateral geniculate nuclei are your optic radiations. And we just saw those in the last slide, so you can see them right here, all these little striations. So all of these are your geniculocalcarine tract. But this little extra swoop that's coming temporally and then hooking back, that there is your Meyer's loop. So here's your Meyer's loop on this side. And then all the rest of these is just your unnamed geniculocalcarine tract. And like we said, the Meyer's loop is carrying the fibers to your lingual gyrus because that came from the inferior retinal fields, superior visual fields, but then when the inferior retinal fields are staying low in Meyer's loop to go to the lower gyrus. And that's why when you're looking at the brain upside down, you can see Meyer's loop literally terminate here in the lingual gyrus, and that's why you can't see these geniculocalcarine tracts terminate, because it's going to be actually more superiorly located deep to our view right here up in the cuneate gyrus. Okay, now we're going to look at our posterior commissure from a few different angles. What's important to know about this is that there are a few visual fibers crossing through this posterior commissure. Nothing we've really covered yet. It's going to be fibers involved in vertical gaze. But just know that the posterior commissure has some crossing fibers involved in the visual system. So let's look at this mid-sagittal section to orient you. Here's anterior, here's posterior. This is the duck area that I like to call it. Here's the pons you can see. So imagine this is a little duck. Its eyeball is the anterior commissure. The optic chiasm is its bill. It's got a little foramen of Monroe, maybe a little ear, something like that. And here's the slope of its head. And you come back, and here's your pineal gland that's poking out into the superior cistern. And just beneath your pineal gland, this small section right here, this is your posterior commissure. It is right above the tectum, or the quadrigeminal plate, with your superior and inferior colliculi. And it also says in the lab manual that the posterior commissure is arching above the entrance of the cerebral aqueduct, which is right here. And then here's your pons and midbrain. And yeah, let's see from a couple different angles. Here's the opposite cross section here. Right here is going to be your posterior commissure. Now, your anterior commissure looks alarmingly similar on a cross-section like this. So you have to know, when you're looking at a cut, how far forward or back you are in the brain. So here you should know you're pretty far back because you have your thalamus present right here, this dark, big thalamus. So if we're far enough back, we can see that. Also, it helps here that you can clearly see that you're already cut into the pons and the brainstem. And you can see the cerebellum. So it's kind of easier to ID the posterior commissure on this than it is the anterior. But let's look on this. We have the stained cross-section. Here is going to be your posterior commissure, and you can tell it's posterior and not anterior because you have your brainstem right here. You have your midbrain with your red nuclei, the substantia nigra here, and the pes pedunculi here. And just to connect some of these pictures, here's your PCOM. Up here's your pineal gland. Why? Because you can look over here and see, oh, here's my posterior commissure and my pineal gland. So it superimposes really nicely on cross-section. And then you can look over from the posterior view. So now if we're looking back at this area like so, we would see we have our quadrigeminal plate, and then right above it, we have our posterior commissure. Okay. So this is my take on the auditory system. We're just going to really quick go through it. Otherwise, you can come back and study it on your own. So we have our cochlea right here. Our inner hair cells and outer hair cells get stimulated and are sending our hearing stuff. So the important one here is the inner hearing cells for hearing. It's going to be picked up with these type 1 spiral ganglion neurons. And type 1 are myelinated, and the cell bodies live in the spiral ganglion No way. Spiral ganglion neuron lives in the spiral ganglion. So here is where your first degree cell bodies are. So they're going to pick up that inner hair cell deflection, going to transport it to your dorsal and ventral cochlear nuclei. And then here is going to be your first synapse. Boom. Now we have second order cell bodies, and what's traveling out is going to be second order. So from the ventral cochlear nucleus, you're going to have bilateral projection of those second order fibers to each superior olivary nucleus. And then another synapse, boom. Now you're in third order. That's your third order cell bodies are living here. And then just a quick rewind, your dorsal cochlear nuclei are going to skip the SONs. Why? Because the dorsal, the dad cochlear nuclei, does not want any sons. He don't want no kids. So he is going to skip right past those bilaterally and just join the lateral lemniscus and skip that part of the pathway. So that said, since there was no synapse here, you're going to have part of the lateral lemniscus that's going to remain second degree fibers. And then also you have a little bonus, just the ipsilateral ventral cochlear nuclei will also send past the SON and contribute another set of second order fibers. So now you can see your lateral lemniscus is going to be covering third order projections from the SON and second order projections from both the DCN and the VCN. So once we're here on the superior olivary nuclei, this is where a lot of your sound discrimination is coming from. So you have, imagine on this diagram, you have your medial superior olivary nucleus and your lateral superior olivary nucleus. Your lateral is going to be sending information back and forth comparing the intensity of sound. So the loudness is going to be lateral, comparing loud. And then your medial is going to be sending information back and forth talking about your phase differences, which is your arrival time of sound. So both of those working together is going to be sound localization. And this area where the superior olivary nuclei are crossing between is called your trapezoid body. And so that trapezoid body is made up of these third order fibers communicating back and forth. And technically part of the trapezoid body is this ventral acoustic stria, which is the VCN's contralateral superior olivary nuclei projection. So you have second and third degree fibers making up this trapezoid body. So then from those superior olivary nuclei, you have your lateral lemnisci traveling up to the ipsilateral inferior colliculi, and then you have another synapse. That's going to turn into your fourth order cell bodies that are going to send fourth order fibers via the inferior brachium to the medial geniculate nuclei. Another synapse, boom, fifth order. Then you're going to finally get to your auditory cortex, which is your Brodmann's area 41-42, your transverse gyri of Heschel. And something important to note is that you can see that once we get past these nuclei, everything's going back and forth, back and forth, back and forth. That said, if you injured just any one part after it's passed through the cochlear nuclei, you're not going to see any hearing loss because there's so much connection between the sides that it's going to be able to make up for that. So you're not going to see profound hearing loss unless you damage the nuclei or earlier. The only problems that you'll have past the cochlear nuclei are going to be difficulty with localization of sound, if anything. So we're going to take a look at these auditory pathways on the lab specimens. So first off, we have our internal acoustic meatus, which of course is where your sound, your vestibulocochlear nerve is traveling through. You actually can't see it, ironically, on this photo very well because here you have your jugular foramen. Your internal acoustic meatus is actually going to be a little bit further up. You can barely see it on this side right here. You can't really see it here, but it's going to be right there on that petrosal portion of the petrosal ridge, petrous ridge of the temporal bone, just right underneath right there. And then we also are just going to point out our auditory cortex real quick. Here are your transverse gyri of Heschel. We've removed this temporal lobe, pulled it up so you can kind of see your insula area almost. Here are your transverse gyri of Heschel, 41 and 42. Okay, now we've got a bunch more cross sections. So since we just talked about the cochlear nuclei, we're going to ID them really fast on here. In the lab manual, I kind of liked the way it said it. It says that you can see the cochlear nuclei sitting on the hips of the open medulla. And when you look at it here, here's your open medulla. You can tell because you've got the big inferior olivary nuclei. And the open medulla, to me, already kind of looked like a pelvis. So if you see, like, here's your hips, where you put your hands on your hips, your pelvis, and then down below where your femurs would go in. So get that visual. So on the hips of the open medulla are where your cochlear nuclei are going to be, which checks out because right here is your cochlear nerve. And here's specifically your vestibular nerve already breaking off from that cochlear nerve. So your cochlear nerve is traveling, and it is going to synapse on those nuclei. So you have ventral and dorsal. So dorsal's easy enough because it's just going to be the most dorsal. So here is where your dorsal cochlear nuclei. Your ventral does look more lateral, so that's why I've included it like that. So here is where your ventral nuclei are going to be. So as long as you remember dorsal is dorsal, then your ventral will just kind of follow around your inferior cerebellar peduncle here. And then we'll again point out some old friends, our lateral lemniscus. We know that the lateral lemniscus, since we just looked at that auditory diagram, is going to be communicating with our inferior colliculus. So here on our midbrain cross-section, we have our inferior colliculi with our lateral lemniscus just hugging right there. And then our brachium of the inferior colliculus, we can see better here. So here's our superior and then our inferior brachium going to our median geniculate nuclei right here. Okay, here we're gonna really quick go to our vestibular system. You can study over this on your own, but just really quick, we've got our vestibular apparatus here. So we're going to use the horizontal crista ampullaris. Your first order fiber is going to have its cell body located in Scarpa's ganglion here. So it's going to travel. We're going to synapse on our vestibular nuclei. And our medial and lateral do different things. So our lateral vestibular nucleus, when it gets signal, it is going to synapse and send out fibers to our ipsilateral extensors. So on the same side. So if you're spinning to the left, that means you're going to activate your left horizontal crista ampullaris. And so it's going to make your lateral vestibular nucleus send out and extend all of your left side. So that way, when you're spinning, you kind of bear down on your left side to go and make that spin. At the same time, for your vestibulo-ocular reflex, that same left turn spin is going to activate your left vestibular apparatus, do your left medial vestibular nucleus, and that's going to send out fibers via the medial longitudinal fasciculus. Ipsilaterally, it's going to inhibit your abducens. And then contralaterally, we're going to activate it. So now that this is inactivated, all of the communication stops here, which means that we're not going to have any activation of our contralateral medial rectus. So what's really important is that it's going to go contralateral to the abducens, and in that abducens, we know from our last exam, has connections to the contralateral to it, which is now going to be ipsilateral to the activated nuclei, medial rectus. The way that I like to look at the vestibulo-ocular reflex is I will stare at the screen. So like stare at my red dot and just stay right here with your eyes and then turn your head to the left. And keep your eyes on this dot. When you turn your head to the left, that is you activating your left vestibular apparatus. And look at what your eyes are doing right now. So your right eye has its lateral rectus engaged because it's out laterally. Your left eye has its medial rectus engaged. So let's see if that checks out. So if we're looking to the left, we can keep looking at my dot right here and turn your head to the left. You should have your ipsilateral eye being your left. We don't want it to be lateral, using our lateral rectus, and it's not. It's only using our medial rectus. And then our contralateral eye, our right eye right now, is definitely using the lateral rectus and not using the medial rectus. So there you go. I know that's probably self-explanatory, but it really helps me to just move my head and keep my eyes on it and then think about the individual muscles and what they're doing. And now the other thing that the vestibular system does, it's got your vestibulospinal tract. So we already kind of showed you this one on this graph, or on the last diagram. But we have that same left-sided vestibule activation. We've got our medial and lateral vestibular nuclei. So we saw that lateral one sending down and activating our ipsilateral extensors, and that's our lateral vestibulospinal tract. But we also have our medial vestibular nucleus. So not only was it sort of helping with our vestibulo-ocular reflex, it also will send bilateral cervical musculature signals via the medial vestibulospinal tract. Okay, so now we got some more cross sections. These are both of the open medulla here. Like we said, here are our hips, and here's our inferior olivary nuclei. Now we're going to locate these vestibular nuclei. We have seen the vestibular area, but we haven't had to distinguish between the 4 nuclei we have to ID now. So here's the easiest way to do it. We have 4 nuclei that we're gonna have to see: superior, lateral, inferior, and medial. It perfectly spells out SLIM, S-L-I-M. So lower, you can see the vestibular nuclei in 2 places. You can see them up in the pons, and you can also see them at the top of the medulla and the open medulla. So the pons is above the medulla. So the pons is where you're going to see the S and the L of SLIM. And then here you're going to see the I and the M. So SL is higher, IM is lower. So here we are in the IM field. So if we have the vestibular nuclei named inferior and medial, inferior is not a very good directional term when you're looking at a cross section, but medial is. So let's ID our vestibular area first. So vestibular is going to be sensory, so let's find our sulcus limitans, and it's going to be lateral to that. So this is our vestibular area, right? So if we were going to think, but in this vestibular area, where would the medial nucleus be? Probably medial, and it is. So this is our medial vestibular nucleus, which leaves left over the inferior. It doesn't make sense in the name, but it's perfect because we know medial is here, so whatever's left over is inferior, and that's going to be right here. So here is your medial and inferior vestibular nuclei. And just to look at it on a different color-stained cross-section, here's our sulcus limitans, here's our medial vestibular nuclei, and here's our inferior vestibular nuclei. Medial, inferior. So now we are up higher. That means we're in the SL of SLIM. So we're going to do the same thing. Superior doesn't really give us much directional sense, but lateral does. So let's ID whereabouts our sulcus limitans is, so about right here. Really good, delineated by this facial nerve arching. Here we have our vestibular area. So we would imagine that the lateral vestibular nucleus is more lateral, and it is. So our lateral vestibular nucleus is right here, while our superior is here. So superior, lateral. Same deal over here. Superior, lateral. Superior, lateral. It's kind of hard to distinguish right here, but I think she'll make it pretty clear if she's going to ask us about that. And then a little shout out to the future. You can see here we've got some cerebellum showing because here's your dentate nucleus interposed and fastigial. Okay, so here we have taste really quick. Taste is fully ipsilateral, so we have our taste bud receptor cells. They are going to be sending their information to our facial, glossopharyngeal, and vagus nerves, so 7, 9, 10. 7, its first degree is going to be in the geniculate ganglion. The glosso's first degree is inferior petrosal, vagus, inferior nodos. Anterior 2/3, posterior 2/3, and then all the rest of your oropharynx. So just like your epiglottis and your tonsillar area, all that is vagus. They're all going to come, and it's still first degree because these are pseudo-unipolar, so we don't have any synapsing until we get to the solitary nucleus. All of these first degree fibers traveling to the solitary nucleus are going to be using the solitary tract. So this right here, these fibers, it's the solitary tract. Boom. It's going to synapse in the gustatory cortex of the solitary nucleus, which is that rostral pole of the solitary nucleus. Now here's where your second degree cell bodies are. Then they're going to send ipsilaterally up the centrotegmental tract to get to the VPM of the thalamus. And then boom, we have another synapse. Third degree is then going to go to our gustatory cortex, which is in the insula. It's in that, really in the anterior insula region. And just again, to note, it's fully ipsilateral. So if we do have any taste deficits in this happening, it's going to be on the ipsilateral half of the tongue. Here, really quick, is olfaction. The thing to remember about the way these fibers end up converging is that we have all these odorant zones on our olfactory epithelium here. And there's not too much rhyme or reason. We just know that they are specifically organized almost to be random. But when we come through our cribriform plate of the ethmoid bone, and we're going towards our olfactory bulb where we have our glomeruli, all of the odorant receptor neurons that are sensitive to the same type of odorant are going to converge on the same glomerulus. So you can see like all of these red fibers are going to end up going to this red glomerulus. And this is where you're going to have your first synapse. So now you're going to have second degree cell bodies here, and those are going to be mitral and tufted cell bodies. And they're going to come up here, and we're going to create a couple of stria. So the main one that's going to go to your olfactory cortex areas, which is right here in this temporal, medial temporal lobe region. We have the piriform cortex, the preamygdala region. We are going to call all of these going there the lateral olfactory stria. So the main communication from the sense of smell from all the way to your cortex is going to be lateral olfactory stria. However, that implies there's a medial olfactory stria, and there is. Because coming off of the olfactory bulb on the way to the cortex, we have our anterior olfactory nuclei, which are located sort of on the medial aspect of this lateral stria. And we don't know too much about it. We just know that for now, we just know that the anterior olivary nuclei are going to send some smell fibers back and forth between the contralateral sides, anterior olivary nucleus. And so this crossover is going to be called your medial olfactory stria. And because it's crossing through the brain, there should be a commissure that lines up with this, and there is. So this is passing through the anterior commissure. So that duck eye that I pointed out earlier in the mid-sagittal section, that is where these fibers are crossing. Okay, clearly I was really creative with this slide, so lab 8. All right, here we hit the cerebellum. So we have folia and fissures, kind of the same way we have gyri and sulci. So each little outcropping is a folia, and then each little indentation between is a fissure. We have 3 major lobes. So we have an anterior lobe, a posterior lobe, and a flocconodular lobe. So the best way to delineate the lobes is with the biggest fissures you can see. The primary fissure here is separating your anterior and posterior lobes. So you also can kind of see a fissure here, and you'd be like, well, why isn't that the main one? I kind of like to see the tree roots here, so when you follow this fissure in, you like hit this tree root here. But in the real primary fissure, you're going to be able to follow this line all the way to the core. So here's your primary. Then you have your posterolateral fissure right here, and that's going to separate your posterior lobe from your flocconodular lobe. And then we have one more pretty major fissure, and that's just going to cut this posterior lobe in half. So right here, you're going to see this is your horizontal fissure, separating the 2 halves of your posterior lobe. Now, when you cut it mid-sagittally like this, you can see that within each lobe, there are some pretty clear lobules, and it's just a smaller little, essentially part of the lobe. Some of these are named. So here we'll start over here. So this is your superior medullary velum, if you remember. So here's your 4th ventricle. This tiny little piece sitting on the superior medullary velum is your lingula. So here's your lingula. This right here of your flocconodular lobe is your nodule. So when you cut mid-sagittally, the part that you can see in the vermal area is what we're going to be naming here. So that's why this is called the nodule of the flocconodular lobe. And then just adjacent to the nodule, we have the uvula. And then just adjacent to the uvula, we have the pyramis. So the uvula is aptly named because it sits between the two cerebellar tonsils. So you can see one tonsil here really nicely. The other one's obviously been cut away. But that's going to help us ID later. So if you ever see a piece of cerebellum in between the two tonsils, just know that that there is the uvula. Now here we have a different way to look at the cerebellum. So let's look really fast. We have the cerebellum. Essentially, this is looking at it from the anterior view. So the brainstem here has been removed. And you can tell because the cerebellar peduncles are what connects the cerebellum to the brainstem. So we have our superior, middle, and inferior cerebellar peduncles. They kind of make a little outward triangle shape when you're trying to connect them in your head. And we've got a couple things to look at here. So imagine on the last slide we had cut it mid-sagittally and we're looking at it like from this side. So we had seen our nodule, so that's where our nodule is right here. And then adjacent to the nodule, you remember, is the uvula. And if we see the uvula, we know that it is between the two tonsils. So here are our cerebellar tonsils pushing in on the uvula. And then we can see our three lobes here from a different view. We've got our anterior lobe up top, posterior lobe below, and then we have our flocconodular lobe, because here's our nodules. That's the vermal part of our flocconodular lobe, and we can see it extending outwards, kind of like a little bird flying with its wings outstretched. So that's our lobes there. And then just a note, the cerebellar tonsils are a part of the posterior lobe. Oh, and then also here, you can see that this fissure right here is separating our flocconodular lobe from our uvula, which is part of the posterior lobe. So this is our posterolateral fissure. Now here we're back to that cross-section of the pons I showed you earlier. We have your three cerebellar nuclei. We've got our dentate, which is our outermost. Dentate looks an awful lot like the inferior olivary nucleus, that's squiggly. Then interposed between the dentate and vestigial is the interposed nucleus. And then our fastigial nuclei right here. And a little throwback to when we just learned our vestibular nuclei with a SLIM acronym. Pons is higher than medulla, so we're at the top half of this word, SLIM. So we should see our superior and lateral vestibular nuclei. Okay, here's a cross-section of the cerebellum, and it's as if we've just sliced it and are looking down on the top of it, is an easy way to think about it. So we've got our dentate nucleus, our interposed nucleus, and our fastigial nuclei. And then here we've got our nodule. Okay, really quick. Here's the cerebellar cortex we were talking about. We have got the three layers of the cortex. We've got molecular, Purkinje, granule. Molecular is most superficial. Purkinje is just one cell layer thick of Purkinje cell bodies. And then the granule layer is our innermost. So when we have information coming in, there's two ways that it comes in, via climbing fibers or mossy fibers. Climbing fibers are only from the inferior olivary nucleus, and they will directly excite whichever deep cerebellar nuclei we're talking about, dentate, interposed, vestigial. It'll directly excite it, and then right after it does that, it continues up, grabs onto the Purkinje cell, and excites it. But the Purkinje cell itself is inhibitory. So it basically, the climbing fiber is going to turn on the deep cerebellar nucleus and then indirectly turn it off through the Purkinje to time your motions. And so then your mossy fiber is coming from everywhere else. It's similar in that it directly activates your deep cerebellar nucleus, but then it's going to take an even more indirect way to go and turn this off, because it's going to synapse with a granule cell, which then will come up and with its parallel fibers excite everything in the molecular layer essentially, and then you'll have that activation of the Purkinje cell to then send that inhibition. And you can see on this side, what also happens with the granule cell is it has a mechanism to turn itself off. So it comes up here, excites this Golgi cell, which inhibits the granule. So we've got timing sequences here, on, off, on, off. And then since we have this middle player, it needs its own off switch. So it's on, off. And then just a little shout out to the stellate and basket cells. This parallel from the granule cell is what's going to excite these, which are inhibitory cells. So that's why you get lateral inhibition, and you're only exciting the one part that you want to at a time. Okay, don't freak out. I don't think it's that bad. I do, but it's okay. Here are two different ways to look at the cerebellar pathways. So that's why it looks so busy. This is the same diagram, just two different visual ways. We're going to start with the archicerebellum. That's going to be your vermis. It's going to be your vermis. So it's your medial most part of your cerebellum. It's going to do mainly your medial motor systems. So you can take a closer look at this on your own, but just a really quick show, most of our input is going to be coming from either directly from our vestibular apparatus, or it's going to come with a quick synapse in the vestibular nuclei. So that's going to hit our flocconodular lobe and vermis, where the archicerebellum is located. And via the Purkinje cells here, like we said, are inhibitory, the nuclei, the deep cerebellar nuclei associated with archicerebellum is fastigial nucleus. And it makes sense because the archicerebellum is the medial most part and the fastigial nucleus is the medial most deep cerebellar nuclei. And so then the point of this all is to then send information back out to the vestibular system. So overarchingly, you're going to get vestibular information, send vestibular information out. And that's going to control your equilibrium, your medial motor system, because if we'll remember, all those medial motor systems we talked about earlier were coming from our, like vestibulospinal tracts and stuff like that. So next, with our paleocerebellum, it's going to communicate with the deep cerebellar nuclei that's just lateral to the vestigial. So we've got our interposed nuclei as a part of this system. So whereas our archicerebellum did all of our medial system, our paleo, our next one outward, is going to do our lateral system. It's going to modulate that. So the way that it's going to do that is it's going to use all of our spinocerebellar fibers that we learned way long ago. And it's going to take information from that and from the cortex, and it's going to end up sending information out to talk to not only our cortex, which then is going to communicate with our spinal cord, which is the goal. It's also going to communicate with our red nucleus to send our rubrospinal tract to do the lateral motor system of that cervical region, so our arms. And then this is going to be all the rest, the motor systems here. So overall, modulate lateral motor system. And then, so our damages here is going to be just typical lateral damage. So distal flexor ataxia, intention tremor is a thing with this. So when you go to grab something or do a sort of movement, you'll develop a tremor there because you're trying to utilize the system that is faulty when you're reaching your limbs out to do something. That's your lateral motor system. Now we're going to end with our neocerebellum. So that's our most lateral, and it's going to use our most lateral deep cerebellar nuclei, our dentate nucleus here. And what that's going to do is sort of feedback and do our really complex skilled movement, helping plan and put together learning skilled movement. So it's kind of like, it's still taking into account your lateral motor system, but it's more compiling everything together. It's kind of like how the primary motor cortex is just doing the movements, but then the premotor cortex is helping plan. This is kind of what this is doing here. So it's going to essentially make a feedback loop, right? So we get information from the cortex and the inferior olivary nucleus, which is showing us, how our motor stuff is doing down below the spinal cord, and our cortex is giving us ideas for what it wants to do. We're going to send it to the dentate nucleus, and then the dentate nucleus is going to send information back out to the cortex and the inferior olivary nucleus in this big circle, just this huge output circle that's going to be modulating your movement. So then damage is going to be like your loss of really skilled motor planning. So movement decomposition is a really good way to say that. But yeah, I'll have this just in the slides for you to review on your own and walk through it really step by step. Okay, really quick basal ganglia talk. Honestly, it's As long as you can just memorize the order in which this goes, it'll be super simple. So cortex is talking to striatum. Striatum talks to GPI, which talks to thalamus, which talks to cortex. That's the direct pathway. You have your indirect pathway, which is the same, except instead of directly striatum to GPI, we have striatum, GPE, subthalamic nucleus, GPI, and then it still follows the same path. The way that I've remembered all these neurotransmitters is just to remember the order. So if I can remember the order of this pathway, I can just write out these words, and then to connect the words, I've memorized this. Yes, no, no, yes. And it's same over here. Yes, no, no, yes. But the indirect pathway is a little longer, so it's got an extra no, yes. And the way that I've made this work, and if it doesn't work for you, that is perfectly fine, but it may help somebody, is yes is going to be excitatory, no is inhibitory. So we remembered all these names of these little nuclei, so we've written these out on our paper. Then we start and we go yes, no, no, yes, no, yes. And then over here it's just yes, no, no, yes, yes, no, no, yes. And then when you draw this on your paper, you now know what every single nuclei uses as a neurotransmitter to communicate. So things to look at here, these substantia nigra, when these are interfering and adding dopamine, that is an elective thing. So typically these are just operating without this input. But then, so this is why our cortex with the indirect pathway is being inhibited when we're at rest. But when we want to initiate movement, our substantia nigra throws in that dopamine to then inhibit this inhibitory pathway and excite the cortex. Same thing over here. This one is just going to extra excite when it wants extra movement. So that's why when you take away dopamine, you injure that. You now have Parkinson's because you're unable to excite the cortex via the dopamine pathway. So you're not excited, you're hypokinetic, you got Parkinson's. As for everything else, I've kind of got it written out here if you want to take a look at it, but one just like easy thing just to keep memorized is subthalamic nucleus. If something injures that, you get hemibolismus on the contralateral side. Okay, now we have the nuclei, the basal ganglia on a cross section. So let's look. We have our caudate nucleus here. And our caudate nucleus is also C-shaped. We just can't see it right now because we would have to look at it from this orientation, kind of looking at it from the side. But it follows kind of how the lateral ventricles are C-shaped. So if you imagine this running back with this lateral ventricle, if we remember, the lateral ventricle has temporal horns, right? So when you see the temporal horn lateral ventricle, boop, right there, this caudate nucleus is still holding on to it, but instead of the caudate nucleus being the lateral wall, the caudate nucleus is the roof of the temporal horn of the lateral ventricle. So now moving on, we have the putamen, globus pallidus externa, interna. We have the claustrum, which is this dark stained piece right here. And now we have to think of everything that separates these nuclei. So we have our big internal capsule, these axons coming down through here. And this is going to be our posterior limb of the internal capsule because we have our thalamus right here. So we can tell that we're posteriorly enough that this is going to start being our posterior limb. And then separating, so if our internal capsule's here, our external capsule is going to come right between the putamen and the claustrum. And then we have another little white matter tract that's coming even further lateral from the claustrum called the extreme capsule because it's on the extreme edge of the brain. And here's your insula. And then in between the putamen and globus pallidus and the globus pallidus externa interna, we have our lamina. So we have our lateral medullary lamina and our medial medullary lamina separating there and there. Okay, now looking at it from a vertical view, here's our lateral ventricles. So we saw the lateral wall of our lateral ventricle was our caudate nucleus. So here's our caudate. Here is going to be our lentiform nucleus, which is our putamen plus our globus pallidus. So that section of 3, you can really ID this well here because the claustrum is always really easy to ID. It's always just a skinny piece of gray matter. So if we have our claustrum here, we know that medial to our claustrum was our putamen. So that's why we've got our lentiform nucleus as a whole here. And then since this is a myelin stain, that's why all of our white matter is dark. So here is our internal capsule. You can see our anterior limb, which is coming between our caudate and our putamen. And then we have the genu, because we got a little bend right here. And then we've got our posterior limb of our internal capsule. Let's see, what did I not go over? Oh, we also have our external and extreme capsules we can see. So if here's our internal capsule, other side, external capsule, then we have claustrum, and then here we have extreme capsule. Okay, now we have another cross section. Let's see if we can tell if we're anterior or posterior. So here we have our lateral ventricles. So we have our caudate nuclei, but this is not the thalamus. This is not what the thalamus looks like. The thalamus is just going to be one blob. This is a lot of stuff going on. And this really illustrates how the caudate nucleus, if we remember from lab, it is supposed to anteriorly in the brain connect with the putamen. And you can see that it does, because here's our putamen wrapping around and connecting with our caudate nucleus. And where they connect is now called the nucleus accumbens. So this little arch here is our nucleus accumbens. And so our putamen kind of reached around to grab caudate, but if you remember, what was sitting right here medial to putamen was our globus pallidus. When we get up this anteriorly, it's just going to be our globus pallidus externa. Our interna kind of develops a little further back in the brain. So if you ever just see one part that you know is globus pallidus but don't see any delineation between externa and interna, you're probably just looking at externa. And then again, we have our claustrum here, that right nucleus right there. Then we have our external capsule, our extreme capsule, and then of course our anterior limb of our internal capsule. Because like I said, we're anterior here, so this is our anterior limb. All right, now we've got a fiber dissection cut of the brain. We're going to talk about the different types of fibers here. So we have associational fibers, which are connecting just different gyri on the same side of the brain. So we have short and long associational fibers. Our short ones are arcuate fibers because they kind of make little U's. So arcuate. So here it is, just coming to and from adjacent gyri. So here's some more, just like that. And then we have our long associational tracts, which we have a superior longitudinal fasciculus, an inferior occipitofrontal, and an uncinate fasciculus. So our superior is going to be superiorly located. We see this big wave right here. This is going to be our superior longitudinal fasciculus. And then we have another one lower, so that's going to be our occipitofrontal because it's connecting occipital lobe to frontal lobe. That's going to be right here. And then we also have the uncinate fasciculus, and that is going to be a part of the inferior occipitofrontal, but it's going to be this U for uncinate. So we got a U right here. That is going to be the part of our inferior occipitofrontal fasciculus. All right, so now we've removed a little more fibers so we can see kind of deeper into the brain. We still have some arcuate fibers that you can just kind of imagine right here, here. And here we have our putamen. So if we had dug out around the putamen and have left it here, can we remember what was covering it on the outside of it? So when you were looking at a cross section, what was out lateral to the putamen? It was going to be the external capsule. So these right here are fibers of the external capsule that you can now see from the side. And then we still have our inferior occipitofrontal fasciculus right here. And our geniculocalcarine tract is actually going to be, our inferior occipitofrontal fasciculus kind of blends in and becomes the geniculocalcarine tract. So when you see, like with that wave coming, it ends up sort of just opening up into becoming the geniculocalcarine tract that's then going to come to the occipital lobe. Okay, so I've illustrated on this slide just how easy it is to, once you've taken out your cerebellum and stuff like this, it's hard to tell how far anterior or posterior you are in the brain. So let's walk through how we would figure that out. if you look at this cross section, you see the lateral ventricles, and you see a big blob right underneath of them, big, round. That's your thalamus. So if you see your thalamus, you're posterior enough that your thalamus exists. So just know that you're farther posterior in this section than you are here. Why? Because here's your lateral ventricles. You can't see a thalamus. In fact, you can see your internal capsule coming all the way in. And there's no lump here that would be stopping it from traveling here. So this is going to be the more anterior cut than this is. And this is also why, when I was saying earlier, like when you look at your putamen, globus pallidus externa, and interna, you find your putamen, here it is. You see your globus pallidus. You can't really delineate externa from interna. That's because this is just externa. Interna comes up a little bit further back. So we've got your putamen, externa, interna. You have that claustrum, internal capsule, external capsule, extreme capsule. And since we're posterior, this is the posterior limb of the internal capsule. And then we also have our cingulum. We covered this in lab. It is another fiber pathway, which aptly named, it is in the cingulate gyrus, which, as we know, is right above the corpus callosum. So here's that. Here's our cingulate gyrus. It's that first gyrus above. And our cingulum is going to be the white matter tract within it. So this black for the myelin stain. These are our cingulum. And then right here, these little white interjections, that's our cingulum right there. Oh, and then also, just to point out, our commissures. So now that we've located, now it's a little easier, which one's posterior and anterior. This is our anterior cut. So here you can see our anterior commissure. And here you can see our posterior commissure. And oh shoot, no way, another one. What are we gonna do? Let's pretend like this isn't kind of giving away that we may be located somewhere specific in the brain. Let's just look at this cross section. We see the lateral ventricles. Below it, ooh, big blob. That's the thalamus. Perfect, we're posterior. We see a commissure. Perfect, that's the posterior commissure. Amazing. Little throwback to earlier, here's our caudate nucleus, and remember what I said about it following the lateral ventricle. Let's not forget the lateral ventricles have a temporal horn, and the roof of it is the tail of the caudate nucleus. Okay, quick commissure point out on the mid-sagittal section. Here's anterior, here's posterior. Let's find our duck. Here's our duck. Here's the bill, here's the head, here's its neck. So the eyeball of the duck is our anterior commissure. Our posterior commissure is going to be just beneath our pineal gland. So here's our pineal gland projecting into the superior cistern. Here's our posterior commissure. It makes a little C for commissure, in case you forgot. And then our hippocampal commissure, you're going to have to consider it more theoretically for now because it's very hard to visualize on a cross section. But obviously it's going to be connecting your two hippocampus or your two hippocampi. And where that is going to be located is the fornix. And so this is your fornix right here. So your hippocampal commissure is technically going to be like right about here. But just remember, I don't think that she would ask you to ID it because it's really not visible. But just know that it's just above the posterior commissure. And then, of course, we have our corpus callosum, which is kind of cut off in this picture. But here you can see the splenium of it. Because if we remember, we have our rostrum. And then our genu, our bend is kind of going like that. And then our body. And then our splenium is that big circle, how it's going to end. And look, really shouting out the commissures here. Because I mean, if this is not going to be tested on, then that's my bad. But just in case, check it out. Let's see if we're anterior or posterior here. So we see the lateral ventricles. Do we see a big thalamus blob? No. We see a lot of mess. So that means that we're more anterior and we can see more of the basal ganglia clearly. So here's our caudate nuclei. Here's our anterior limb of the internal capsule. Then we've got putamen, globus pallidus externa, globus pallidus interna. Oh, we also have our nucleus accumbens kind of connecting, because just a little bit we can see our caudate connecting with our putamen, nucleus accumbens. And dang, what's this big white matter tract? Oh, look, it's our anterior commissure. Crazy. And look down here, you can also see that is our optic chiasm. And now let's move over to this cross-section. This already, you're like, oh, boom, that's the midbrain. Well, if you can see that and you're on a brain cross-section, that must mean you're posterior enough to see that. So here's your big red nuclei, looking real pretty. We've got our pes pedunculi, substantia nigra, and we have a commissure right here. And it also happens to be just below this circle, which is our pineal gland. So this must be our posterior commissure. And of course, here's our lateral ventricles. We know we're posterior now, so boom, we hit a big blob. Thalamus. And a little bonus shout out, what is part of the thalamus are your medial and lateral geniculate nuclei of the thalamus. So if you see two little blobs, I'd imagine this one's the medial and this one is the lateral geniculate nuclei. So there and there. All right, and so that is all of this review. If you have any questions, come find me or text me, email me, what have you. If just feel free to look through those diagrams that I've included in the slides if you want to go a little more in depth. I know I covered them pretty quickly, but it was just to get a quick little overview.